Yes, actually. Uh,
Okay. Any other
but
questions?
it is difficult. It is a it is a challenging the estimation of alpha value because alpha is a parameter of the estimator. So we have to build an estimator to choose the estimator, which is little challenging.
Okay. Thank you very much. Okay. Let's go to the second presentation. It's my professor to, it's my pleasure to introduce Professor Naila. Professor Naila is the head of proteomics at QU Health at Qatar University. She is a leading glycation researcher from the omics technology and an expert in the development of diagnostic algorithm. She is pursuing research on glyosylate, one inducer for prevention, rehearsal, and treatment for type 2 diabetes. She has over 147 publications and 9 patents, receiving over 15,300 citations and 8 index 67, and is ranked in the top 2% scientists globally for citation of her work. So the title of her article is Application of Machine Learning to Develop Diagnostic Algorithm Using Metabolic Markers of Protein Glycation Oxidation and Nitration in Oxygen. Let's welcome Professor Naila.
Is it okay if I just sit down?
Yes.
Right. Thank you very much. Well, my name is Professor Naila Rabbani, and I am in College of Medicine in Qatar University, and I'm very happy to be here talking about application of machine learning to develop diagnostic algorithm using metabolomic markers of protein glycation, oxidation, and nitration in autism. So I would like to briefly discuss autism and the challenges of its diagnosis. Autism spectrum disorder is a developmental disorder in children, and the prevalence of it is increasing, and the difficulty is the diagnosis of, of the condition. This is because of the high variability and heterogeneity of the symptoms, makes diagnosis of ASD difficult and uncertain, particularly at the early stages of development. And this condition is believed to be affecting over 60 million people worldwide. Current diagnosis is based on observations and interviews to assess behavioral and communication characteristics by a child development expert. By the time parents can have spot the difference, it's in their children of or spot the symptoms at the age of 9 and 12 months, but the time it takes to refer to the clinical or child development experts takes another 4 years, and further assessment, and then the diagnosis happen, and the care and support is provided. By the time it is provided, it is quite late. Average age is about 4 to 5 years before the diagnosis is made. So we have been trying to provide a solution and developing blood tests for autism, because in early intervention can produce remission by counseling or cognitive restructuring. It can help, but because of the delays in diagnosis, for some children, it's too late. So if we can validate this blood test, it could and make improve access to diagnosis to support early intervention and care, which could produce remission. So our study design and methods, we measure trace level of amino acid in the plasma using a technology called ageomics. We measure modified and unmodified amino acids. So totally 40 different type of amino acids are measured in the plasma protein or in the plasma filtrate. Glycation is a process which can be disturbed in metabolism, and oxidation and nitration can happen if there is immunity or other conditions. So we totally measure about 40 different biomarkers in blood, and we correct these multiple analyte measurement and for to prevent any false discovery rate. And then, of course, we use the machine learning to using ensemble and sport vector machine with twofold cross validation and repeated 10 times to develop the algorithm. So, as I mentioned, the methodology is the gold standard stable isotopic analysis, LCMSMS, for markers of protein damage, and the analysis time is only 30 minutes. We take non fasting blood sample and to measure these markers using LCMSMS. So this is a gold standard. You can have it in any lab. You will have the same results because it is corrected with internal standards. And then we use our proprietary diagnostic algorithm to uh, present the probability of either presence or absence of the condition. And the diagnosis can be done within 7 days compared to over 4 years. So the initial, our discovery phase, we found that there were these markers, carboxymethyl lysine and carboxymethyl arginine, increased significantly. And these markers are both CML and CMA are formed in protein modification by glyoxal. Which is formed in lipid peroxidation. So this is a reactive metabolite, which is coming from the lipid peroxidation pathway. So there may be disturbance of lipid peroxidation in ASD. And secondly, we found that the another marker called dityrosine content of plasma protein is increased about 87% in ASD. And dityrosine is mainly formed by protein modification by dual oxidase, and duox is increased in response to microbial challenge to gut and lung mucosal immunity. So there is likely increased duox mediated protein modification in plasma and interstitial fluid in ASD. So these are the interpretation of our data. And of course, you can't have one biomarker can provides you diagnosis. So we use machine learning, sport vector machine learning, to have all these 40 biomarkers and develop the diagnostic algorithm, and found that the four features emerged as critical for accuracy of the classification. As I mentioned before, CML, CMA, and dityrosine were increased significantly and even passed the bond for any correction, and 3-deoxyhydroglucosone, hydroimidazolone, it is a glycation marker, was decreased in ASD. And as you can see, the accuracy was 88%, and area under the rock was 0.94. So here is the our results on developing. We developed three different type of algorithms from the same. It was plasma protein, where amino acids were digested, or the protein was digested to the component amino acids, and then use LCMSMS to measure these markers. Then we also can measure proteolysis product, free amino acids in plasma filtrate and urine filtrate. So we use all these compartments to develop diagnostic algorithm, and the protein plasma algorithm was the best one, providing 0.92 sensitivity and 0.84 specificity, and diagnostic odd ratio was 68. Plasma amino acids, only two of these, CML and CMA, gave um, reasonable, what you call it, algorithm, but not, of course, good. So likelihood ratio, positive likelihood ratio for plasma protein was 5.7, and the negative likelihood ratio was 0.097. What this mean is that negative likelihood ratio below zero point 0.1 tells you very convincing evidence of the absence of condition. So what we have, this diagnostic algorithm was very good at telling a child who do not have autism and about moderate evidence that there is a presence of autism. So now the challenge is to validate this diagnostic algorithm. The work I did discovery phase was done in College of Medicine, Warwick University, and then I and my colleague we relocated to Qatar, and I was very lucky that I have access to three different cohort of autism. So called Doha cohort one was Qatar Biomedical Research Institute collaboration, and there they have tests at cohort one had 144 children cases and control of age 2 to 5 years, and then the second Doha cohort come from the Baraka study from Sidra Medicine Hospital. It's Women and Children Hospital. And there I have access of 126 children, and these are the age of 5 to 12 years. And that was the age of my discovery cohort, which I did work with the University of Bologna in Italy and to develop the initial or training the algorithm. The third, I also developed collaboration with the Dr. Yolanda Diego from Malaga Institute in Spain to access these third cohort of 66 children cases in control with ages range between 1 and 5 years. So this is has been challenge as we were discussing yesterday, that if you develop an algorithm using the cohort with 5 to 12 years old, then testing that with the younger age is going to be a problem. So, our ongoing clinical validation, our aim has been to survey of protein damage markers in plasma of children with ASD and normal development in Qatar, providing data for blood test development in Qatari cohort of different AGEs. And the second aim is to improve our diagnostic algorithms and validate the method in an independent subject cohort. So validate the diagnostic algorithm with independent set. And I'm also developing using different method, but not me, the collaborator in computer science. I've been asking them that you have to come up with something better, which is really specific for this type of data. And they come up with this ANFIS using an adaptive neurofuzzy inference system, mathematical model to train the algorithm. So I will just present you the preliminary results. So the survey of protein damage markers in plasma of children with ASD in normal. So as you can see, I am showing you the selective level of amino acid and modified amino acid of the our original discovery phase. And we found there was not huge difference in the level we found in children of younger age and in Qatar. So that this means that the plasma amino acids and glycated amino acid levels in Qatari cohort were similar to those of the Bologna cohort, and diagnostic algorithm may be applicable to population of differing age and ethnicity. The ongoing this validation, we have so far measured protein damage markers in 104 children out of 144 samples analyzed to date, and algorithm developed with SVM. And bear this mind that these are younger children, and the algorithm we developed initially was of older children. So they were a little bit, I ask my collaborator from he is Kashif Rajput from University of Birmingham, but based in Dubai, to just test it for me anyway. How does it look like? So it wasn't too bad, but it wasn't too great either. So he did say that you can't test with the younger children, so we have to work on that. So any suggestion will be gratefully appreciated. But what we did find, the four feature critical for accuracy in the discovery cohort are changed similarly in validation cohort. So we found that even in these younger children, CML, CMA, and diatyrosine are increased, and 3-deoxyhydroimidazolone is decreased in ASD in this younger cohort. So this gives us some confidence. So further analysis is ongoing, and we are exploring classification performance of age-corrected algorithm development and influence of confounders. Now, this is then I have collaborated with the computer scientist in Qatar University, Dr. Awais, and her PhD student use all my plasma filtrate data and use this ANFIS to develop diagnostic algorithm. And she did great job. But this time we have 4 amino acid and 5 damaged amino acid. If you remember, in the first one we developed, we only have 2 features, CML and CMA, and that gave about 67% sensitivity. But with these 4 amino acid and 5 damaged amino acids using the Bologna cohort, it come up accuracy was 97%, and sensitivity specificity was increased 86% sensitivity and 99% specificity. And what was so important and exciting, that the positive likelihood ratio was 85, and the negative likelihood ratio kind of was not as good, but positive mean that it was conclusive, convincing evidence that the child have autism. And the diagnostic odd ratio increased from 9 to 625. So we are exploring that, and hopefully we will be able to validate to that same sensitivity and specificity, which I have seen many studies. It failed validation. So it would be nice to know what we can do to prevent failure in this. So why does Ageomic have advantage over other omics technologies for diagnosis of autism? I would like to say that what our protein damage markers are normally not recycled. I mean, these are not normal metabolomes, normally recycled or further metabolized. It never accumulate in different condition. But what we measure, we look at the damage to the amino acid, and these accumulate and do not recycle. So that's one reason why we see the difference in our data in cases and control. So what we found, our biomarkers are, as I've shown you, interpretation is really mechanistically involved in the condition. And the recent study, transcriptomic studies of post-mortem brain in ASD found increased expression of chaperone gene for increased protein folding. This may reflect a response to increased damaged and misfolded protein. So which I reported in this Ageomics. So we are looking at this damaged protein, and we are finding in the brain of autism the the gene which proteolyze the damaged protein is increased. So to summarize, a simple blood test for autism has been developed based on assay of plasma amino acid. Assay time is 30 minutes. Discovery phase gave positive likelihood ratio 5.7 and negative 0.097. What it means is that a strong convincing evidence for absence of autism if the negative likelihood ratio is below zero point 0.1. Validation and independent test set cohorts are ongoing, and SVM was used in discovery algorithm development. AMPHIS may be superior, studies are ongoing, and diagnostic tests based on protein damage analytes in plasma may be reflecting a protein folding response in the brain in autism. With that, I would like to acknowledge my team and the key collaborators, and I would like to thank you for listening. Thank you.
Professor Naila, for the input into this. This is very meaningful because early diagnosis can help to the prevention and the better treatment for autism.
Mhm.
Yeah. So any question to Professor Naila? Yes, please.
Sorry? False positive. So as you could, we did have the F value or that. So it was there was maybe 2 or 3 false positive, but the first data was very small. It was a discovery phase, but now we are having almost 300 cases in control. So we will know how it validates the algorithm.
And I also want to add some remarks, particularly I remember the last slide in PowerPoint, and this is amazing the result on the validation check. Or maybe like the slide before this slide, and a table shows the specificity area on the curve. So I think it's not that it's quite it's not easy to get such a beautiful result.
Yeah.
I also read some other papers, and in their paper, particularly on the validation set, they counted with such a high likelihood for result. This is impressive.
I agree, and I am praying that the validation give the same, but my collaborator is saying it doesn't look very good. He have to do all these cleaning and confounders and stuff. So any other models, any other mathematical program which might help, or if somebody is developing for specifically certain type of data set, that would be great to know because this protein damage markers can help a lot. Right.
Any other question? Yes.
Well, these children, as I said, the younger one, which I have studied from even 6 months onward, so we see the same type of changes. But the older children, these changes were highly significant. That 40 analyte, it was corrected for bone franny and still were significant. But the younger children, you can see the trend is there, but not as highly significant. So at that point, if we do intervene, then it could prevent. So you could see as the age is growing, these protein damage markers are increasing.
Thank you.
Thank you.
Professor Naila. Okay, now let's go into